In this lesson, we will examine the issue of cellular adaptations. Normal cells are usually in equilibrium with their environment. When this equilibrium changes, the cells adapt and undergo cellular and metabolic alterations in response to these influences. Typical stresses may include such things as increased or decreased stimulation caused by hormones or growth factors, increased or decreased workload, decreased blood flow, or exposure to abnormal materials such as toxins. When the environment changes, cells can alter their metabolism as well as their morphology in an attempt to adapt. However, if they are not able to adapt to the stresses, or if the nature of the injurious stimulus is too severe, the cells may not be able to survive, and the result is cell injury and cell death. Cell injury will be covered in a subsequent lesson. How cells respond to stress is associated with the type, strength, and duration of the stress, but it's also dependent upon the nature of the cells themselves. Cells can be classified into three general types: labile, stable, and permanent, according to their status in the cell cycle. Labile cells are continuously dividing and thus are continuously progressing through the cell cycle. Examples of labile cells include cells of the epidermis, on your skin for instance, and cells lining your gastrointestinal tract. Quiescent or stable cells are not normally dividing, so they have left the cell cycle. However, if they are stressed, they can re-enter the cell cycle and divide. Hepatocytes are an example of cells that are not normally dividing in situ, but when stressed, they can divide. Permanent cells have left the cell cycle permanently and are not able to divide even if they're stressed. Examples of permanent cells include neurons and cardiac myocytes. Although recent evidence suggests that in some situations, neurons and cardiac myocytes may actually be able to divide, although this is probably from the stem cell pool rather than adult differentiated cells themselves dividing. So it's a combination of the cell type and the environmental stress or change that influences how the tissue will respond to a stimulus. We have categorized these responses into four basic processes: atrophy, hypertrophy, hyperplasia, and metaplasia. These processes are better demonstrated here in this diagram. In this figure, the normal state is depicted as a single layer of cells sitting on top of a basement membrane. If we stress the system and the cells undergo atrophy, that is, the individual cells will lose their cellular components and they will shrink. In the second example, hypertrophy, we see that the cells, when they undergo a stress, the cells must work harder, and this necessitates an increased functional capacity and results in increased cell size. In this case, the cells are not able to divide, so they can only respond to the stress by getting bigger. The third example is hyperplasia. In this case, the cells are able to divide, so in response to a stress, the cells proliferate. This leads to an increased number of cells and subsequently an increase in the size of the entire tissue or organ. The next example is a process called metaplasia. This process involves a change from one differentiated cell type to another differentiated cell type. And the final example is dysplasia. This involves a transition to a pre-neoplastic state, and this process will be discussed later when we describe changes resulting in neoplasia. So if we start looking at these various processes and start with atrophy. Atrophy, as we said, is the shrinkage in size of the cells by loss of structural components. In this cartoon, we see that the normal cells on the left shrink. The components of the individual cells get smaller, and the cells themselves get smaller. The types of things that can cause atrophy can be listed as things such as decreased workload, loss of innervation, diminished blood supply, inadequate nutrition, or loss of endocrine stimulation. And there are probably others as well, but these are the main ones. But let's look at a clinical example of atrophy. And for this case, we look at disuse atrophy of skeletal muscle. Anybody who's broken a leg and had to wear a cast knows that your leg muscle, since you can't walk and use that leg, the leg is not used, so you end up with disuse atrophy. If we took a muscle biopsy of this patient and looked at cross sections under the microscope, we can see in this H&E stained section that some of the muscle cells are fairly normal in size and configuration, as shown here in the by the green arrows. However, if you look at this section, there are a bunch of cells that are really small and sort of abnormal shaped. This is a process of atrophy. So here at the ends of the blue arrows, and you see in many cells throughout this tissue section, the cells are small. So this is an example of atrophy of skeletal muscle caused by disuse atrophy, or the broken ankle caused this patient not to use the muscle as much as normal. Another example of atrophy is what we call senile atrophy. The brain on the right is smaller than the normal brain on the left. And this is an aging process and often caused by a disease process, such as Alzheimer's disease, that leads to senile atrophy of the brain. Another example of atrophy is in the kidney, and we call this hydronephrosis. The kidney on the left has undergone atrophy. What's happened is a stone has been lodged in the ureter, and this blocks the flow of urine. So as the urine, you get a backlog of urine, and as the renal pelvis fills with urine, it causes pressure from the inside of the kidney. So the kidney almost looks like a balloon with the renal pelvis full of urine. And this is under high pressure because the kidney keeps trying to make urine and filling up the renal pelvis. Eventually, of course, the pressure gets high enough that the kidney doesn't work anymore. But as you can see from this gross section from an autopsy specimen, the kidney has undergone atrophy because of that pressure of the urine in the renal pelvis. Also in this section, you can see the other kidney, the contralateral kidney. This kidney has undergone hypertrophy because it's had to work harder and increase workload because the other kidney was damaged and was not working properly. So to summarize, atrophy is the shrinkage in the size of the cell due to loss of structural components. And this results in a decrease in the size of the organ or tissue. What about hypertrophy? As we said, hypertrophy is increased size of the individual cells, and this results in increased size of the organ. Again, if we look at a cartoon, the normal cells on the left, and hypertrophy, there aren't an increased number of cells. However, each individual cell gets bigger. So the cellular components are increased, thus giving you a bigger cell, hypertrophy of the cell. What are some things that can cause hypertrophy? Well, we sort of break these into two groups. Physiologic hypertrophy is an example of normal physiologic process leading to hypertrophy. The classic one is when you have hormonal stimulation during pregnancy, which causes the uterus to get big. So this would be an example of physiologic hypertrophy. Pathologic hypertrophy is where you have some sort of a pathologic state that causes the tissue to work harder, and thus you get hypertrophy. One of the classic examples where an organ has to work harder is the heart. And this is when you have an increased functional demand leading to hypertrophy of the left ventricle. And the classic example of why this would occur is either systemic hypertension or a valvular stenosis, for instance, an aortic valvular stenosis. Here's an autopsy specimen, and we're looking right down onto the top of the heart. The aorta has been cut away, and we're looking at the aortic valve. Now, the aortic valve should have 3 separate leaflets. In this case, the leaflets are thickened, and the leaflets are fused such that the hole or the orifice of this valve is very small. So, because the orifice is small from calcific and stenosis of the aortic valve, the blood, the left ventricle has to work really hard to pump the blood through that calcified aorta. So as the blood goes into the left ventricle has to squeeze really hard to force the blood through that stenotic aortic valve. And this is what results. On the right, we see what we call left ventricular hypertrophy as compared to a normal heart on the left. In the hypertrophied heart, the wall is significantly thickened because that left ventricle has had to work so hard to pump the blood through that stenotic valve. And thus the muscles are bigger and the left ventricle is bigger as compared to the left ventricular free wall of a normal-sized heart. This is obviously caused by hypertrophy of each individual muscle cell. And here on this histology section, you can see the hypertrophied cell on the right as compared to the size of the normal cell on the left. So this is left ventricular hypertrophy, a pathologic type of hypertrophy caused by aortic stenosis. Another example, in this case a physiologic hypertrophy, as we mentioned, a postpartum uterus. The uterus is bigger than normal because it's had to hypertrophy or increase in size because of carrying the fetus. So this is physiologic hypertrophy. And then one last example of hypertrophy, again sort of a classic case. In this case, we're looking at an autopsy specimen. We're looking at the inside of the aorta, and the aorta has a lot of atherosclerosis. And in fact, in the renal artery has been partially occluded because of atherosclerosis from the aorta into the renal artery. And you can see that this kidney has atrophied because of decreased blood flow to that kidney. And once again, just like we saw before, the other kidney has hypertrophied as a reaction to the decreased functional capacity of the contralateral atrophied kidney. So compensatory hypertrophy due to renal artery stenosis leading to atrophy of the other kidney. So to summarize, with hypertrophy we see an increased size of the cells, and because of this increased size of the cells, we see an increased size of the tissue and organ. Hyperplasia, as we said, is an increase in the number of cells in an organ or tissue. And again, if we look at normal cells, they're put under stress, they are able to proliferate, so they proliferate in order to respond to that stress. Just like with hypertrophy and hyperplasia, there are what we call physiologic hyperplasia and pathologic hyperplasia. Again, the example of physiologic hyperplasia is hormone-induced cell proliferation, and this is most prevalent in, for instance, the the ductular epithelial cells in the breast that are stimulated to produce milk, and as they're stimulated by hormones, they proliferate, giving you a physiologic hypertrophy. Pathologic physiologic hyperplasia because of increased proliferation of cells. Pathologic hyperplasia, we see an example of viral-induced hyperplasia. Papillomaviruses produce warts by hyperplasia of the epidermal cells. And the other example of pathologic hyperplasia is hyperplasia of the prostate brought on by excessive hormonal stimulation. This is a classic example at an autopsy section of a prostate of an older gentleman with prostatic hyperplasia. And what we see on this cross-section at autopsy are nodules of proliferating tissue, because this is hyperplasia. And these nodules, as you can see, are bulging here on the surface, the cut surface of the autopsy specimen. If we do a histology section of one of these nodules, we see that the ducts, the prostate ducts, contain an increased number of epithelial cells. So this is an example of hyperplasia. There are so many cells that they're piling up on one another, and as shown by these arrows, in the upper left, they've almost completely obliterated that duct from proliferation of hyperplastic cells. And we also see areas where there's infolding of the duct, of the epithelium, just because there's so many cells and they're proliferating so much. So, to summarize, hyperplasia is an increased number of cells in an organ or tissue. And metaplasia, as we mentioned, is a reversible change in which one differentiated cell type is replaced by another cell type. Now, one of the classic examples of this is when ciliated columnar epithelium of the bronchus or trachea are replaced by squamous epithelium, and this often happens in smokers, chronic smokers. The irritation of the cigarette smoke causes the epithelial layer to change from one differentiated cell type, the columnar cells, to another differentiated cell type, and that is squamous epithelium. But one of the things to remember here is that it's not actually the differentiated cell itself that dedifferentiates and redifferentiates into a squamous epithelial cell. Instead, what's happening is a signal is produced that tells the stem cells, the basal cells along the basement membrane of the epithelial lining, to change their gene program, and instead of maturing into ciliated columnar epithelia, they instead mature into squamous epithelial cells. So here again in this cartoon, to demonstrate that, on the left-hand side is the normal situation in which you have ciliated columnar epithelium in the bronchus or trachea and stem cells that are constantly dividing and replenishing that epithelial layer. Well, if you damage these cells from, for example, cigarette smoke, what it does is it sends a signal to those stem cells that instead of maturing into columnar cells, they mature into squamous epithelial cells, and thus causes squamous metaplasia. As you can imagine, squamous epithelial cells are much more robust, and they're much better at protecting the body from the toxins in the cigarette smoke. So here's a histology section of the bronchus of a smoker, and we see on the left by the blue arrow, here are the ciliated columnar epithelial cells that you would expect throughout the entire bronchial tree. However, these are replaced by an area of squamous metaplasia, as shown here by the green arrow. And again, this is caused by a signaling pathway thought to be related to vitamin A, and it causes the progenitor cells or the stem cells to mature into squamous cells instead of ciliated columnar cells. Now, there's another example of squamous metaplasia can be seen in the kidney. This is an autopsy specimen where we found a kidney stone. That kidney stone was in the renal pelvis, and it was, as you can see, it's very rough. So it was irritating the epithelial lining of the renal pelvis. Now, as you recall, the renal pelvis should be lined by simple transitional epithelium. However, with the stone in there, the stone is rubbing on this epithelial layer and causing injury. So here's a histology section of that renal pelvis after we had removed the stone. And you can see over on the right-hand side, as shown by the green arrow, that here's an area of, looks sort of like a transitional epithelial layer. It's not too thick. It's maybe a little thicker than normal, but it's moderately thickened. And you also notice that in this area, there's a tremendous amount of inflammatory cells. So that stone, that renal stone, has irritated the tissue. And we see the transitional epithelium with some areas of hyperplasia because it's slightly thickened, and then the inflammation as shown by the blue arrow. And over on the left-hand side, as shown by the red arrow, we see that the transitional epithelial layer has been completely replaced by a layer of squamous cells. And so this is the area that we would call squamous metaplasia. If we look at this at higher power, you can see that the basal layer, there's a lot of cells along that basal layer that are proliferating and maturing into squamous epithelial cells instead of forming a normal transitional cell layer. So, to summarize, metaplasia is a reversible change in which one differentiated cell type, be it epithelial or mesenchymal, is replaced by another cell type. So, as we've seen, cellular adaptations take a normal cell that's in homeostasis with its environment, and because of some stressful situation, that cell adapts. And as we mentioned, the types of adaptation are either atrophy, hypertrophy, hyperplasia, or metaplasia. And what we hope is that the stress that the cells are able to adapt to that stress and function, the tissue functions normally. However, sometimes if the cells aren't able to adapt, or if the stress is too severe, you can end up with cell injury, and that will be the topic of a subsequent session. So this is the end of the Cellular Adaptations lecture. I encourage you to look over the cases that are described in the interactive pathology laboratory related to cellular adaptations.